Good day. In this lecture, we will be discussing about the mitochondria and the chloroplast. So, the membrane systems of the bacteria, the mitochondria, and the chloroplast are very similar in such a way that we can say that they are related. So, for a bacterium, you have your it actually has two membranes. You have an inner membrane and an outer membrane system enclosing the cytoplasm. And if we look at the mitochondria and the chloroplast, you will see that both organelles, both cellular organelles also have double membrane systems. So you have an inner and an outer membrane. Although they are, for the mitochondria and the chloroplast, they are different. And of course, the chemical makeup there are variations in the chemical makeups of the membranes of these three structures. The fact that they are similar sparks assumptions that perhaps they might be related somehow. Actually, this is one of the premise of the endosymbiotic theory. Although as of now it's still a theory, but again, the endosymbiotic theory postulates that the mitochondrion was previously a bacteria that was engulfed and absorbed by the cell, but instead of the engulfing cell destroying it, the cell uses it to generate its ATP. So basically, that's the idea for the endosymbiotic theory. Now, the mitochondria and the chloroplast, both of them have roles in ATP, in energy generation and conversion. And they do it through chemiosmotic coupling. So in chemiosmotic coupling, we have here several stages. So the first stage in chemiosmotic coupling is that you are harnessing the energy from either sunlight or the food to generate an electrochemical gradient. So in the case of the chloroplast, the source of energy is from sunlight. And they harness the photons from the sun to generate your electrochemical gradient. In each case, you have hydronium ion gradient. For the case of the mitochondria, it's the energy from the food. So the glucose, for instance, they oxidize glucose. The energy from the oxidation of glucose is used to generate this electrochemical gradient. And once the gradient has been generated, here comes the real the ATP harvesting portion of the chemiosmotic coupling, and why it's called chemiosmotic coupling. So if you remember from the previous lectures that we have active transport that is generated by coupling it with an electrochemical gradient. The same goes for your mitochondria and the chloroplast in this chemiosmotic coupling mechanism. But instead of doing an active transport, the energy from the electrochemical gradient is actually used to synthesize ATP. So if you remember, ATP or adenosine triphosphate is the energy currency of your cell. So this is it's actually used to create usable energy for the cell. So what makes it possible is the ATP synthase. So it is embedded in the membrane. So you have the lipid bilayer of the membrane, and it allows the passage of hydronium ion, but with atoll. So for every rotation, basically, because it's very similar to a rotor machine. So for every rotation of the hydronium ion that passes through here, it will generate a certain amount of ATP. So let's look first at the mitochondria, or the mitochondrion, singular term. So the mitochondrion or the mitochondria occupy 20% of the cytoplasmic volume of a eukaryotic cell. It doesn't mean that the mitochondria is actually very large, but rather there are many mitochondria in a cell. It's generally small, but as it occupies 20% of the cytoplasmic volume, that means there are many mitochondria. So basically, if you are thinking that your cell has a mitochondrion, it's not. Your cell has many mitochondria. So they are often depicted as short bacterium-like bodies with a diameter of 0.5 to 1 micrometer, but they are in fact remarkably dynamic and plastic. So they move about in the cell, constantly changing shape, dividing, and fusing with one another. So without the mitochondria, present-day animal cells would have had to generate all of their ATP through anaerobic glycolysis. And tissues with high energy demand, such as the cardiac muscles and even our skeletal muscles, they have large amounts of mitochondria in their localized in these muscle cells because again they require the energy that this mitochondria will generate. So the mitochondria, looking at the structure, what is this? The striking feature of the mitochondria is its double membrane system, so the inner and the outer membrane. So shown here is the electron micrograph of your mitochondria. So these lines here are actually the cristae in the inner membrane. So basically, your inner membrane is larger in terms of the surface area than the outer membrane. So that's why it forms folds, it's folded up inside. So here is a tomographic slice through, a 3D image generated from your electron micrograph, and this is a artist sketch. So the outer membrane, it envelopes your inner boundary membrane. But since the inner membrane has a larger surface area, so it's folded up inside. So you have these squiggle lines. These are the folds of the inner membrane. And then the matrix is the interior of the membrane. Now we call the folds cristae, and the space within it the crista space. So the mitochondria, the crystal space of the mitochondria is actually very important for the function of the ATP generating function of the mitochondria. You have here, if you're going back to the respiratory respiration, basically the metabolic pathway of respiration. So the first step in respiration, if you remember, glycolysis occurs outside of the mitochondria. It actually happens in the cytoplasm. But the byproducts, the products of the respiration, they are shuttled to the citric acid cycle, and the citric acid cycle occurs inside the matrix of the mitochondria. So they enter the mitochondria. Not only the glycolysis, the pyruvate from glycolysis, which is the end product from glycolysis, enters, but also fatty acids from fatty acid metabolism. They enter the mitochondria, and this is where the citric acid cycle occurs. And then they generate, this is where the oxidation of the carbons in the pyruvate happens, and as well as in the fatty acids. And it will generate many electron carriers. Now, the third step happens in the intermembrane space. The third step, which is the electron transport chain, where electrons is passed around a series of molecules embedded in the inner mitochondrial membrane, which in turn, for every electron that is passed around by these proteins, they shuttle hydronium ions to the intermembrane space. And because of the crista and the folds, your intermembrane space, this portion of the intermembrane space becomes highly acidic. And that is what fuels the chemiosmotic gradient of your for ATP synthesis. So the chemiosmotic process couples the oxidation energy to ATP production. So in the process of your oxidative phosphorylation, so the inner mitochondrial membrane serves as a device that changes one form of chemical bond energy to another. Thereby, you convert a major part of energy of NADH oxidation into the phosphate bond of ATP. So you have here your oxidative phosphorylation. So NADH, the oxidation of NADH generates ATP. And then this is a chemiosmotic process, which is through the oxidative phosphorylation is basically a substrate level phosphorylation. But here, the chemiosmotic regulation. So another process, these are the two different processes. So oxidative phosphorylation is a substrate level phosphorylation. That means you have a substrate and use the energy of the substrate to generate the production of ATP. Another way to generate ATP, and this one is much more efficient, is to couple it with a chemiosmotic gradient. So the chemiosmotic gradient generated by the protons or the hydronium ions, we call it the proton motive force. So you have here a higher concentration of hydronium ions in the intermembrane space. Although if you look at the actual pH, the actual concentration, you can see that it's actually quite low. The difference is really quite just a little. So it's about 0.7, a pH 0.7. So 7.2 to 7.9. So it's relatively not that far in terms of a pH difference, but because of the cristae, the hydronium ions become concentrated in the in certain portions of the cristae where the ATP synthase occur. So the proton motive force here is actually a twofold force. You have your membrane potential because protons, these are passively charged ions, so it generates electrical charge. And you have, of course, the chemical gradient. So concentration gradient plus the potential, the electrical gradient. So this you form your electrochemical gradient, which is a proton motive force. So it's basically a voltage and concentration. So acting together, it generates ATP production. So how does this proton motive force is generated? It is generated through the action of these proton pumps. So the proton pumps, they for every the electron carriers, this comes from the respiration, the citric acid cycle. These are electron carriers for the citric acid cycle. So they deliver the electron through here and they pass around the electron. So I won't really delve too much on which goes where, but for every passage, you have an expulsion of protons to the intermembrane space. And then you have your ATP synthase at the end of the cristae that harness the generated protons, the proton motive force, to generate your ATP. So this is your ATP synthase. So here is the actual image of your ATP synthase. So it's actually a very large molecule, and it's actually the rotor ring is actually spinning around as the hydrogen ions enter. So it spins around like a Ferris wheel. So the crista, as I said before, the crista is important for the mitochondria because the pH is 7.4, 7.9, and the outer membrane has pores that allows passage of the ions. But in order to actually fully harness the proton motive force, you want to concentrate the hydronium ions on a certain regions, and that is where the creases or the cristae in the mitochondria comes into play. So as you can see here, the red dots are the you have your hydrogen ions. So in the red dots of the hydrogen ions, they do not really diffuse out of the cristae space, but rather they tend to be concentrated on the dead end where the ATP synthase are located. So this allows a more efficient ATP production despite having a small proton gradient between the cytosol and the matrix. So for the case of the chloroplast, so chloroplast, on the other hand, they contain core machinery for the photosynthetic pathway. So chloroplasts use also chemiosmotic mechanisms to carry out energy interconversion in much the same way that the mitochondria do. So they are much larger than the mitochondria, but they are organized on similar principles. Like your mitochondria, they also have highly permeable outer membrane, a much less permeable inner membrane, in which your membrane transport proteins are embedded, and a narrow intermembrane space in between. So chloroplasts are localized in areas where photosynthesis occurs. So in the leaf, for instance, so you have lots of chloroplasts here. So basically, it gives the green color for the plants. In much the same way as the mitochondria gives the reddish color to the tissues, especially the muscle tissue. So you basically, if you remember, you have the red meat and the white meat. So it has something to do, a portion of the color is due to the presence of the mitochondria in these cells. The other portion, the other reason for the color is the presence of the uh, myoglobins in these muscle cells, which are storage for oxygen. So they have the heme group that gives it the vibrant red color. But anyway, so you have here your chloroplast localization, so the green pigment from the chlorophyll in the chloroplast. So chloroplasts are generally larger than the mitochondria. So you can see this is mitochondrion, this is the chloroplast. So your mitochondria has a matrix as the interior space, but for the chloroplast, it's slightly different because although it has double membrane system, it also has a membrane system in the interior. So you have thylakoids. These are pancake-like structures that stuck together in the interior of the mitochondria. The grana is the interior of the thylakoids, and then you have the stroma, which is the space on the interior of your chloroplast. So chloroplasts capture energy from sunlight and uses it to fix carbon. We won't delve too much on the metabolic pathway of photosynthesis here, but we will only talk about how these how chloroplasts can capture energy from the sun. So in this case, it's a photosynthetic electron transfer reaction. We call it light reaction because it requires light. So it captures energy from the sun in the form of the photon, so generate ATP and reduce NADPH, nicotinamide adenine diphosphate, H, because these reduced forms. And then the second part is the carbon fixation reactions. They use the ATP and this is NADPH to synthesize metabolites and sugars. So similar to your to the chemiosmotic gradient, this also generates a chemiosmotic gradient. Your light, once the light passes through your photosynthetic pathways, it triggers movement of electrons. For every movement, there is a movement of proton in the thylakoid space. And this proton, you will generate a proton motive force, which the ATP synthase at the end will also utilize to create ATP. And the ATP will be used in carbon fixation. So let's look first at the light harvesting reaction. So this is called a Z scheme. So, well, basically similar to letter Z in a way, if you look at it like that. So the thylakoid of plants and cyanobacteria, they contain two photosystems. So you have photosystem 1 and photosystem 2. So they work in a series type of reaction. The first one will be photosystem 2. So each of the photosystem 1 and 2 reaction centers receives excitation energy from its own set of tightly associated antenna complexes known as LHC1 and LHC2, light harvesting complexes. Light harvesting complex 1 and light harvesting complex 2 by resonance energy transfer. So it's the same way that basically how microwave how it's the reverse way of how metals glow when they are hot. So when sunlight the photons touches, or rather when the photons strikes the light harvesting antenna complex, so it will excite the atoms and it will induce the excitation of energy. So excitation of electrons from lower orbit will become excited and jump to the next quanta, and then it will also trigger a movement of the electrons. So the ultimate source of electrons here is the water. They split water. So this is the water splitting section. So they split the water. It becomes excited. It gathers electrons. The ultimate source is water. And it actually undergoes series of reactions, ending with the final electron acceptor. Rather, the electron acceptor for the light harvesting reaction is NADPH. Now, as I said, photosystem 2 goes first before photosystem 1. So here is how it actually looks at when we look at the redox potential. Because again, we are talking here about how electrons are transferred between photosystems. So you have redox potentials. And why photosystem 2 is the first one, the photosystem 2 basically is the one that harvests, rather that splits the water as the ultimate source of electron. Photosystem 2 goes first, followed by photosystem 1. And why 2 comes before 1 is because photosystem 1 was discovered first before photosystem 2. So that's why they are called the 2 comes before 1. So photosystem 2 here passes electrons derived from water to photosystem 1. So photosystem 2 has a lower redox potential than your photosystem 1. So it goes first. So it then passes, the photosystem 1 then passes them to NAD plus through ferroredoxine, NADP plus reductase. So basically the source is NADPH. So the net electron flow through the photosystem is from water to NADP plus to produce NADPH. And for every passage, for every jumping or passing of electrons, there is a proton movement where they generate your electrochemical gradient. In a similar manner, how the mitochondria generates electron gradient, the proton motive force or the proton gradient, for every movement of the electrons, there's a movement of hydronium ion. So in this way, similar movement of electrons, there's a movement of hydronium ion. So they use the proton gradient to create ATP. So here is your photosystems. Photosystem 2 is passing to photosystem 1, and then final the acceptor for the light reaction is NADP plus. But in the way that they pass around the electrons, they generate hydronium ions is also moved from the interior to the thylakoid space, and they use the concentration of the hydronium ions to generate ATP. So that is how your chloroplast actually works. So comparing them, the H plus concentrations and the arrangement of ATP synthase in the mitochondria and the chloroplast. So the mitochondria, you will see the cristae at the dead ends of your cristae are the ATP synthase generated because the hydronium ions tend to become concentrated here. Now, for the chloroplasts, they tend to become distributed rather uniformly in the thylakoid membrane. This is because the proton gradient was, or rather is being enclosed inside the thylakoid space. So there is really not much worry, or rather, because the hydronium ion, the low pH gradient, if you look at the pH gradient of this one, the low pH gradient is already contained inside the thylakoid. The plants or the chloroplast is actually free to create a much lower pH than the mitochondria. And so they do not really have to worry much about the concentration or the positioning of your ATP synthase. So they are positioned much rather uniformly in the mitochondrion, in the thylakoid space. In contrast, the mitochondrion, you have here a lower difference in the pH between the intermembrane space and the matrix. And because of this lower pH difference, the cristae, the positioning of the ATP synthase is actually important. So the cristae tends to focus the hydronium ions on this side of the rather they tend to concentrate the hydronium ions near where the ATP synthase are. And the reason why they cannot really generate a very low pH in the intermembrane space is because the outer membrane of the mitochondria is very permeable. It's permeable also to hydronium ion, and they do not want this hydronium ion to leak into the cytosol of the cell because that would be detrimental. It could be dangerous for the cell because again, this is a low pH. It's an acidic environment. It can cause damage to the actual cell. So they cannot really lower the pH. Unlike in the case of the chloroplast, the thylakoid space is able to fully contain the low pH of this region. That is why they can lower the pH. So basically, this a comparison of your hydronium ion concentrations and the arrangement of the mitochondria. So that's it for the mitochondria and the chloroplast. So the reference for this book, for these two organelles is from the Molecular Biology of the Cell by Bruce Alberts and Company.